Welcome to the Wright Science Daily Revision Boost. In today's Biology Blast, we're going to look at meiosis and genetic crosses. Hopefully we remember that normal human body cells have 46 chromosomes as they are diploid cells. So this means they've got two sets of each chromosome. The sex cells, so the sperm and the egg or the ova, have 23 chromosomes. They are haploid cells, so they only have one set of each chromosome. What happens in fertilization is two haploid gametes join together to form the diploid cell called a zygote. And that zygote then divides many times by mitosis to produce the new organism. Do remember that every organism produced by sexual reproduction has a unique genome, except for identical twins. When we're thinking about how these gametes are made, they're made by meiosis. Make sure you do spell that right. Meiosis produces 4 haploid cells from one diploid parent cell. And in humans, this will take place in either the ovaries or the testes. The first thing that has to happen in meiosis is the chromosomes are copied through DNA replication. Once that's occurred, the chromosomes line up along the equator in pairs, and then the chromosomes are pulled to the opposite poles, and then the cell divides by cytokinesis. Now hopefully that's all very familiar because it's just mitosis, which we've already learned. However, the key thing to remember here is that there is a second stage. So the chromosomes then line up along the equator, they're pulled in half so a single copy of each chromosome moves to the poles, and then each cell divides into 2 to produce our 4 haploid cells. So just remember it's mitosis followed by another line up along the equator, pulled to the poles, divides in 2. We know that organisms that reproduce sexually have variation, and that variation is caused by 3 things. First, random assortment of the chromosomes at the equator, so that means different ones end up at the poles. Secondly, crossing over of the chromosomes where little bits are traded. And finally, the random fertilization of an egg with a sperm. So when it comes to our variation, you've got 2 copies of every gene, one from the mother and one from the father. So those genes may be the same or they could be different. So a couple of key terms we need to know here. An allele is the different form of a gene. So you've got a gene for eye colour, however the alleles could be for blue eyes or brown eyes. And then phenotype is the characteristic that we see, the observable characteristic there. And most phenotypes are caused by the influence of multiple genes rather than just one single one. When we're talking about alleles, they could either be dominant or recessive. Dominant alleles are ones that will be expressed if either 1 or 2 of them are present, and we show those with a capital letter when we're doing our diagrams. And the recessive allele is the characteristic that will only be expressed if both alleles are recessive, and we use a lowercase letter to represent those. So I've given you a couple of examples at the bottom there. Dominant characteristics, brown eyes, dark hair. Recessive characteristics would be blonde hair and blue eyes. 4 more key terms to know. Genotype is the combination of alleles present in an organism. Homozygous dominant means we've got 2 copies of the same dominant allele present. Homozygous recessive is 2 copies of the same recessive allele present. And heterozygous means that we've got 2 different alleles, so 1 dominant, 1 recessive. Don't forget, when we're asked to work out a genetic cross on the exam paper, you're going to draw a Punnett square, which I've shown you in the bottom right there. So we've got 6 key steps to this. State the phenotype of both parents, state the genotype of both parents, and state the gametes for each parent. Then we fill in our Punnett square, making sure that we state the phenotype for each offspring, and then we finally put down the percentage of each genotype present. So what we have there covers all of the bases possible. Final thing for our biology blast today is just to remind you that your sex is determined by the X and Y chromosomes. So males will have 1 X, 1 Y, and females 2 X chromosomes. So if it asks you which chromosomes would determine someone is male, don't just write down Y because you've forgotten the X chromosome that comes from the mother. So male XY, female XX. Today's chemistry check is looking at some of those key calculations. So the first one we need to be able to do is the percentage yield. So percentage yield is actual yield divided by theoretical yield times by 100 because it's a percentage. So the first thing we need to do is calculate the relative formula masses. Then we calculate the sum of the relative formula masses using the balanced equation, so your relative formula mass times the number of moles. Then we calculate the theoretical yield using the little equation here, mass of limiting reactant divided by the sum of the relative formula masses for the reactant times the sum of the relative formula masses for all products. And then we can calculate the percentage yield using our theoretical yield and the actual yield given in the question. A quick check you can do on your exam to make sure you've got the percentage yield right is make sure it's somewhere between 0 and 100%. If it's not between 0 and 100, you've gone wrong. Flip the numbers round is most likely where you've gone wrong. So what we find is that the reasons our percentage yield would be less than 100 is because the reactants may react in a different way than expected in the air, the reaction may not go to completion, or we may lose some of the product. The second calculation we need to know is atom economy. So atom economy is a measure of how many atoms in our reactants actually end up in the desired product. So something with a low atom economy means only a few atoms in the reactant end up in our desired product, whereas if it's got a high atom economy, then lots of the atoms in our reactant end up in our desired product. So to work out atom economy, it's the sum of the relative formula masses of the desired product divided by the sum of the relative formula masses for all products times 100 because it's a percentage again. And what we find here is that the higher the atom economy of a process, the more sustainable and greener the process is. For those of you doing higher tier chemistry, we need to know how we choose a reaction pathway. So various factors that we will consider when selecting a reaction pathway are the yield of the product, the atom economy, its rate of reaction, any byproducts being formed, and the equilibrium position if we're talking about a reversible reaction. When we talk about a byproduct, these are chemicals which are formed in a reaction in addition to our desired product. Now, some of these could be useful and therefore could also be sold, but others may be toxic or of little use, so they're the ones we want to try to minimize. The last calculation we need to know is about concentration. So it's only for the higher tier on combined science. And what we need to do, first of all, is remember to convert between centimetres cubed, which they're probably going to give you in the question, to decimetres cubed. So to convert centimetres to decimetres cubed, we just divide by 1000. And make sure you do that because any volumes need to be in dm cubed. So concentration in grams per decimetre cubed, we just do that by the mass of our solute in grams divided by the volume of the solution in decimetres cubed. If you're doing GCSE chemistry, you also need to know how to work out the concentration in mole per dm cubed. And all we do there is the amount of the solute in moles divided by the volume of the solution in dm cubed. In today's physics prep, we're going to finish off that last bit of P5 for the separate GCSE physics course. First thing we need to know then is how to draw ray diagrams. Hopefully something we've done at Key Stage 3, but we use ray diagrams to show reflection and refraction. 5 key points to remember. 1. Use a ruler. 2. Draw lines to represent the rays. 3. Draw the normal at 90 degrees to the surface at the point that the ray hits it. 4. Measure the angles between the normal and the rays. And 5. Add arrows to show their direction. So I've given you examples of reflection at the top and refraction at the bottom. For those of you doing the higher tier physics, you need to know what happens with refraction. So we know sound waves are refracted when they change speed. And what we actually see happening here is that when light enters a denser medium at an angle, it's going to slow down and bend towards the normal. So we're still seeing that refraction, that slowing down and the change in the velocity there. The greater the difference in density, then the greater the change in the direction of the ray will be. So one of the things we can actually do is use our knowledge of the behaviour of waves to actually solve a problem. So because the Earth is curved, it means that transmitting waves across large distances is problematic to us. So in order to send radio waves a long distance, we reflect them from the ionosphere, which is a layer in the atmosphere. High frequency radio waves or microwaves, we use those for satellite communications, and they pass up into the actual orbit where the satellite is held, and then they get bounced back down. One thing to bear in mind when we're trying to work out what's going to happen to waves is that it's all down to the wavelength as to what's going to happen to them. So something like a wall will transmit radio waves and microwaves, but will absorb the visible light and the ultraviolet. And the atmosphere is very useful to us because it absorbs X-rays and gamma rays, which means we're protected from them, which is very important, otherwise life would not exist on Earth. Next thing we need to know about are lenses. So we've got two types of lens. Convex or converging lenses refract the rays to a principal focus or focal point. So these are the ones we use in magnifiers, and I've given you the ray diagram there on the left. When we're talking about the focal length, that's the distance from the optical centre to the principal focus. Second type of lens, the concave or diverging lenses, spread light out, as you can see from the ray diagram on the right. And in this case, the focal length is the distance from the virtual focus to the optical centre. One of the problems of sight that we looked at in our biology is looking at how we can correct short sight and long sight. Hopefully we remember short sight means distant objects are blurry, so the rays are focusing in front of the retina. So in order to fix this, we use a concave lens which spreads the rays out so that they will actually focus on the retina. If you've got long sight, so near objects are blurry, the rays are focusing behind the retina, so we use a convex lens to refract the rays in so that they do focus on the retina. If you were to be asked to draw a ray diagram with lenses, then there's 3 steps that we need to follow. First one, we draw a ray from the top of the object to the lens parallel to the principal axis. Secondly, we draw a ray from the lens through the focal point. And then finally, we draw a ray from the top of the object through the centre of the lens. Last thing we need to know about lenses are some of the uses of convex and concave lenses. So a convex lens would be used in a magnifying glass, and it would give us a virtual magnified upright image. We'd also use a convex lens in a camera, which gives us a real diminished inverted image, and in a projector, a real magnified inverted image. Whereas a concave lens is used in spy holes and coach rear windows, and both give us a virtual diminished upright image. Last thing we're looking at in our physics prep today is light and colour. Hopefully we know that as light passes through a prism, then each frequency of light travels at a different speed. As a result, each frequency of light is refracted by a different amount, with the higher frequencies showing more refraction. And as a result, we get this dispersion of the different colours of our spectrum, allowing us to see those different colours on the other side. If we think about the eye, then we know that the retina is sensitive to red, green and blue frequencies because we've got those cone cells that will detect them. So the receptor cells detect the frequencies and the brain interprets the signals sent to make perceived colour in our mind, so we know what we're looking at. If we consider filters, if we were to place a red filter in front of white light, then a red filter absorbs all frequencies except for the red. So the red light's transmitted, but if we place 2 different colour filters, then all light is absorbed, so objects look black. If we just consider what happens when we look at a green ball in different light. If we use white light, the ball looks green because green light is reflected and all other wavelengths are absorbed. But if we put that same green ball in red light, it would look black because there's no green wavelength to be reflected and the red is absorbed, so it just looks black. In terms of reflection, what we find is that if we're thinking about mirrors, then these allow you to see a sharp image as reflection is specular or regular, whereas paper won't give you an image that you can see that's sharp because it's got diffuse scattering, which is the 2 diagrams you can see in the middle. What we do find is that light can be scattered from particles. So milk appears white as the particles scatter all wavelengths, and ink appears black as the particles absorb all wavelengths. And the molecules in our atmosphere are very small, so only scatter light with short wavelengths, which happens to be blue, hence our sky looks blue on Earth. Now we've had a quick recap, then head on over to socrative.com and test your knowledge on the topics from today. The room name you need to enter is Wright 2662. Good luck! As always, if there are bits that we've gone over today in our Daily Booster that you're not certain of, head on over to the playlists and double check the detailed videos. So for biology it's B5, for chemistry C5, and for physics P5. And remember, the physics is only for the separate sciences. The chemistry is mainly separate sciences with one bit on concentration for just the higher tier folks.